Hello everyone. Today's topic is methods of sterilization in plant pathology. Sterilization is the most basic and essential activity in a plant pathology lab. There are microbes in every possible places around us. Microbes are there in air, soil, water, and even the food we eat, the water we drink, or the materials we use. So, in a laboratory setup, when we decide to work with one or only a few microbes, we need to exclude the remaining ones. Here, sterilization is the process that we follow. When we want to make a thing or material free of microbes, sterilization is the process we follow. So, what is sterilization? It means killing all sorts of organisms. In plant pathology labs, while working with some microbes or pathogens of interest, other microbes or pathogens need to be excluded. And this can be done through a process called sterilization, and it is the basic and everyday affair in a plant pathology lab. There are many methods that are required to be followed in various experiments. Before we go deep into the topic, we have a term called disinfection. This term disinfection is different from sterilization. The term sterilization means all sorts of microorganisms will be killed, whereas disinfection means reducing or eliminating harmful microorganisms, mainly the vegetative forms, not the spore forms. So, sterilization ensures 100% freeness from microorganisms, whereas disinfection means reduction of most of the microorganisms or killing or elimination of most of the microorganisms. Sterilization is done usually with high heat and pressure or high heat only, radiation, filtration, or chemical. Disinfection is usually done with chemicals. Now, we also see another term called pasteurization, which is not basically a sterilization. We know now that sterilization means killing all sorts of microorganisms. Pasteurization does not ensure complete killing of all microbes from an object. This process was named after Louis Pasteur. It is mainly used for milk. This is done in two ways. So, milk can be heated at 63 degrees Celsius for 30 minutes to get most of the microbes in the milk killed so that the spoilage of the milk is delayed, or the milk is not spoiled because most of the harmful microbes will be killed through the process by heating at 63 degrees Celsius for 30 minutes. This can also be achieved alternatively by heating at 72 degrees Celsius for 15 seconds. High temperature short time method, HTST. So, pasteurization is basically a process to eliminate most of the microbes from milk. So, some microbes will be still there in the milk after pasteurization because pasteurization does not kill all the microbes in milk. We also see another term called Tyndallization. The process is named after John Tyndall. It is a method of sterilization to kill vegetative and heat resistant spores of microbes. In plant pathology, we use this process to sterilize samples of soil. How this is done? Sterilization is done repeatedly, so it is also known as fractional sterilization. Let us see here briefly how Tyndallization is done. For example, we have a soil sample. We take the soil sample in a conical flask or in a heat resistant plastic bag, polypropylene bag. Now, the first step is heating of substance at 100 degrees Celsius. Moist it for 30 minutes. So this is the job done once in a day. After that, the substance, so in our case it is a soil sample, is incubated. The same soil sample will be heated again at 100 degrees Celsius. Moist heat. We'll get to see what is moist heat and dry heat in the upcoming slides. So here we heat the substance at 100 degrees Celsius for 30 minutes again in the second day, and we incubate after the heating is done. So incubation is done at room temperature. Again on the third day, the substance is heated at 100 degrees Celsius for 30 minutes. So a process of heating repeatedly over a period of 3 days can ensure killing of microbes from the soil sample completely. So why do we need heating 3 times? So look at the expected events at the right side. So when we heat the soil sample at 100 degrees Celsius for 30 minutes, it kills the vegetative cells of the microbes present in soil sample. When the soil sample is incubated, since the sample was exposed to moist heat, the moisture moistens the soil, and the microbes which are there in the form of resting spores, they germinate during incubation period. Now second day we heat the sample again, and the germinated spores in the soil sample are killed in the second heating in day 2. Soil sample is incubated again, so more resting spores which are there in the soil will be germinating, and on the third day again heating is done and germinated spores are killed. So repeated heating and cooling kills all the microbes in the soil, and it is done over a period of 3 days. We know that soil is the abode of or the habitat of microbes. Most of the microbes, soil is a rich source of microbes where we see vegetative as well as spore forms. In order to ensure complete killing of microbes in the soil, we have to follow Tyndallization process. Simple sterilization process that is usually done in one day in one step cannot sterilize soil. So we have to follow Tyndallization. Now coming to the methods of sterilization. The methods can be grouped into heat treatment. Under heat treatment we have two types of heat, moist heat and dry heat. So moist heat treatment is done in an equipment called autoclave. Dry heat treatment is done in one equipment called hot air oven. Then we have a number of methods in the second point: red heat sterilization, flaming, and incineration. These are all dry heat treatment only. Then third point, filtration. Fourth, radiation. And the last point, chemical method of sterilization. We have some other methods applied for sterilizing a substance like sound waves, and also under pressure. So those things will not be discussed here in this video. We will focus on these few methods of sterilization which are mostly done in plant pathology lab. Moist heat sterilization. It is usually done in an equipment called autoclave that functions with the same principle as pressure cooker. In this equipment, there will be water which will be boiled through electricity or in oven. Water will produce steam on boiling, and that steam under pressure will sterilize the materials which will be kept inside the autoclave. This equipment, autoclave, is used to sterilize culture media like PDA, nutrient agar, or any other media that are used in plant pathology experiments, and water. So water can be sterilized using autoclave. So what conditions do you need for autoclaving? The temperature of 121 degrees Celsius and pressure of 15 pound per square inch for 15 minutes will be required to ensure complete killing of microbes from the materials that we put inside the autoclave. So this temperature, pressure and duration must be there for autoclaving. We also see a relationship between temperature and pressure. If our temperature is 107 degrees Celsius, then the pressure developed inside the autoclave is 0.35 kg per centimeter square or 5 pound per square inch. So here in this table we see a relationship between temperature and pressure. And it was found that when the temperature is 121 degrees Celsius and the pressure is 1.05 kg per centimeter square or 15 pound per square inch, then it ensures sterilization. So autoclaving works with moist heat. Now this moist heat and dry heat both can be used for sterilization, but there are some differences. So this difference also reflects the differences between autoclave and hot air oven in their functioning. When moist heat has more penetrating power because there is steam, dry heat has less penetrating power. Moist heat or autoclave requires less time for sterilization. It is only 15 minutes, whereas dry heat requires more time for sterilization, 1 to 2 hours. Temperature required for sterilization in moist heat or autoclave is 121 degrees Celsius, whereas that in hot air oven is 160 to 180 degrees Celsius. In the autoclave, killing of microbes occur due to hydrolytic damage of the microbes, whereas in the hot air oven, killing of microbes occurs due to oxidation of microbes or burning of microbes. Now coming to a question that many students have in their mind. They believe that boiling of water can achieve sterilization. Can we just boil water to achieve sterilization? In one word, the answer is no. Because boiling of water occurs at 100 degrees Celsius. This temperature is not enough to kill all microbes. Remember, we discussed just now that in autoclave temperature is 121 degrees Celsius with a pressure of 15 pound per square inch. Now when we boil water that occurs only at 100 degrees Celsius, certain bacterial spores can survive that boiling temperature. So boiling of water does not ensure sterilization, but certainly most of the microbes in vegetative form present in water will be killed. Dry heat sterilization. So this is done in an equipment called hot air oven. So it is electrically powered and we sterilize glassware like Petri dishes and other materials, oil, powder, etc. So all the glassware that we use in the plant pathology lab are sterilized in hot air oven. So here the air inside the oven gets heated and the heat is retained for some hours, and therefore the microbes present in the glassware or any other material put inside will be killed by burning or oxidation. Here also we have a temperature and duration relationship for hot air oven. So as the temperature goes up, the duration also gets lessened. If the temperature is 120 degrees Celsius, then we have to keep the materials inside the equipment for 8 hours to ensure sterilization. If our temperature is 140 degrees Celsius, then the duration will be 3 hours. At a temperature of 160 degrees Celsius, the duration is 1 hour. And if the temperature is further high at 180 degrees Celsius, then the duration required for sterilization is 20 minutes. Most of the researchers they follow the third temperature duration relationship, that is 160 degrees Celsius for 1 hour. There are certain precautions to be used for hot air oven, like don't sterilize bottles with plastic lids, don't wrap petri dishes with paper, don't pack hot air oven with materials. You should keep some space inside the hot air oven so that the hot air can circulate freely in all the corners or places and come in contact with your materials. So the hot air oven should not be fully packed. In that case, if your hot air oven is fully packed, you may not get your materials sterilized properly. And after the materials are sterilized in hot air oven, we should not open the hot air oven immediately. We should allow to get it cooled down to the room temperature, and then you can open the hot air oven. Next is red heat sterilization. It is a sterilization process where inoculation needle or loop are held in the flame, spirit lamp, or Bunsen burner till they turn red. Especially the tip part of the needle or loop, along with the along with some stretch of the top part of the needle or loop. So the needle part here in this illustration, you can see a part of the inoculation needle or loop is red colored. So this part, which is made up of wire, can be made red hot by holding it in the flame, and this can be done more than once to ensure complete sterilization. So we do red heat sterilization once, then take the needle away from the flame so that it cools down and comes back to its original color. Then we take it again to the flame so that it becomes red hot again. So by doing this repeatedly 2 or 3 times, we ensure that this needle is fully sterilized. So any microbe present on the needle part, the wire, will be completely killed by burning in the flame. So this is called red heat sterilization, mostly used for inoculation needle or loop. Then flaming, it is done for metallic objects uh, to burn microbes or possible contaminants on the objects. Here, the object that is to be flamed is dipped in ethanol or spirit before flaming. It does not ensure a complete sterilization as in case of red heat sterilization. Flaming is done for tools like forceps that cannot be made red hot, that can only be flamed after dipping in ethanol or spirit. So it ensures elimination of most of the microbes through flame, through a combination of flame and ethanol or spirit. Next comes incineration. It is a method of sterilization that is used for waste before disposal, resulting in considerable reduction in the volume of waste. As incineration turns the waste into a completely dried content or ash. It is done in incinerator. Mainly, this incineration is done to avoid release of laboratory grown pathogens into the environment like soil, water, air, and prevent spread of disease. In plant pathology lab as well, we should strictly follow this incineration. In many labs, this is not done. So, a student or researcher, after working with a pathogen, bacterial or fungal pathogen in the lab, which is multiplied in the lab in the petri dishes or test tubes, and when the glassware, petri dishes or test tubes are cleaned, the microbes are thrown in the dustbin. So, the microbes which have multiplied manifold in the lab now go to the dustbin, and then from there they can spread to all possible places and create a health hazard, or can also pose a threat to crops because they land in soil or water or in the air. So, in all possible manners, they can spread and create problems later. So, after a microbe is worked with, a pathogen is worked with, it should be disposed well. So, the media where the microbes grew should be incinerated so that the microbes get killed, and then the waste, which is reduced in content because of incineration, should be thrown in the dustbin. So, this should be followed to reduce the spread of microorganisms in the environment. Next comes infrared radiation. So, the materials to be sterilized are passed through a tunnel at a predetermined speed with source of infrared radiation that gets absorbed by the materials and produces enough heat to sterilize the material. So it is a radiation produces heat and the material gets sterilized. So it is a radiation method for sterilizing substances. Then comes filtration. It's a unique method of sterilization. No heat is involved here. And one very important aspect of filtration is microorganisms are not killed in this technique. They are just separated or removed. By definition, we know that sterilization is killing all sorts of microorganisms. So here filtration is a method of sterilization, but it does not involve killing of microbes. The microbes are simply removed from the materials through a filter paper. So the filtration, since heat is not involved in this technique, this filtration method of sterilization is suitable for heat sensitive materials. So the microorganisms and particles of similar size are removed at the same time. In this technique, we have membrane filters with tiny pores about 0.22 micron are used to prevent the passage of bacteria and fungal spores. The diameter of bacterial cell is 0.3 micron. So if the size of the pore in the filter is 0.22, the bacterial cells will not be able to pass the membrane filters, and the fungal spores will be larger than the bacteria. So these membrane filters with the pore size of 0.22 micron will exclude both bacteria and fungi. So in the filtrate, we will only get sterilized material free from microbes, free from bacteria and fungal spores that can be used further for other works. Now we have another method of sterilization, radiation. We have two types of radiations: non-ionizing radiation like UV rays with a wavelength of 200 to 400 nanometer. UV rays are routinely used in plant pathology lab. We have UV lamp in the laminar flow cabinet. These UV rays are non-ionizing. They have no penetration power. So the surface where UV rays fall, microbes get killed there only, on the surface. Now UV light has a wavelength, a range of wavelength 200 to 400 nanometer, but UV light of 260 nanometer, or to be more specific 254 nanometer wavelength, has the highest microbicidal or bactericidal activity. Now in this wavelength of UV light 254 nanometer, the DNA of the microorganisms shows maximum absorbance. At this wavelength of 254 nanometer, UV light causes formation of thymine dimers in the DNA. So the replication of DNA is affected in the microorganisms and the microbes are killed. And we use these UV rays routinely in the laminar flow cabinet to sterilize the cabinet floor. We also have ionizing radiation like X-ray and gamma rays that can be used for sterilization. These are ionizing radiations because they are high energy radiation. They cause ionization of substances, produces hydroxyl radical, superoxide ion and hydrogen peroxide. And these products are toxic as they oxidize the cellular components in the microbes that get eventually killed. Now we have a chemical method of sterilization. Based on the nature of sterilizing agents, we have gaseous sterilant like ethylene oxide, formaldehyde, nitrogen dioxide, ozone, etc. We also have liquid sterilant like hydrogen peroxide, glutaraldehyde, hypochlorite, and ethanol. Now, here we have a list of some items which are sterilized in plant pathology lab, and the methods followed. For media, nutrient media and water, the method followed is autoclaving or moist heat sterilization. For glassware, hot air oven or dry heat sterilization. For inoculation loop and needle, red heat sterilization. For antibiotic or fungicide solution or any heat sensitive aqueous solution, filtration. For floor surface of laminar chamber, ethanol 70% and UV light. So with this the topic ends here. Thank you very much.